Hello, this is Jasmine Trigg and I'm a scientist at Sartorius. Today I'll be discussing a poster that was previously presented at the American Association for Cancer Research, or AACR, conference. And this was titled Enhanced Phenotypic Screening, a kinetic multiplexed approach to analysing drug effects using live cell analysis. Phenotypic screening is a valuable tool for analysing the multitude of effects that drug treatments have on cells. Traditionally, image-based screening uses fixed cells that are stained with reagents at a pre-selected endpoint. Using our live cell imaging-based technique, this combines kinetic label-free readouts with non-perturbing fluorescence reporters to enable holistic, information-rich quantification of compound effects. Mechanisms of cell death were examined by correlative analysis of the total dead cell population, caspase active and annexin-5 positive cells. The perturbance of cell cycle and AKT signaling pathways were identified using reporter lines, and in these instances, the combination of fluorescence data with kinetic profiles and morphological information yielded additional insights into compound mechanism. So shown here is an overview of the phenotypic screening workflow. So A549 adenocarcinoma cells were treated with a library of over 800 FDA-approved drugs. And images were acquired in the InKsight live cell analysis system every 2 hours for 4 days. Cell growth and viability were measured using label-free readouts of confluence and dead cell count, while cell cycle, cell death, and AKT pathways were investigated using multiplexed fluorescence readouts. So for assay validation and the label-free cytotoxicity analysis, we measured cytotoxicity using the label-free InKsight AI Cell Health Analysis software module, and this allows us to report the percentage of dead cells per well. The live dead classification was confirmed by examining the phase contrast images overlaid with cell segmentation and the classification masks. So this classification is indicated by the color, and you can see an example of this in the image here. So we have live cells segmented in green and dead cells segmented in red. If we look at this microplate view, this shows the kinetic data for the 96 well plates, including the control compounds. And we use this to visualize the kinetic data for the 80 different test compounds and examining this percentage of cell death over the assay time course. We also looked at assay robustness and we calculated the Z prime value using control data. This showed that the Z prime increased over time and reached a maximal plateau between 48 and 72 hours. If we look at the endpoint data shown here in D, this displays the percentage of cell death for all compounds at 72 hours. And this really enables rapid identification of the cytotoxic hits within the screen. The information on compound mechanism of action was then elucidated using correlative analysis. So the compounds we were looking at were separated into either cytotoxic, cytostatic, and non-effective groups. And we did this by examining their cell growth, for which we use the percentage confluence readout, as well as their viability, using the percentage of live cells readout at 72 hours. So where the cells displayed high growth and viability, the compounds had minimal effect. Where they had high viability of low growth, this indicated that compounds are cytostatic, as they're preventing the cells from proliferating without inducing cell death. Where in the cases where viability and growth are both low, this indicates that the compounds are cytotoxic. So the compounds displaying the cytotoxic are shown in this magenta box, and cytostatic is shown in the blue box. And we were able to separate these from those that had no effect on cell growth, as shown in the teal box. The relationship between total cell death and apoptotic death was also examined. So the majority of the compounds inducing cell death were also positive for both annexin-5 exposure and caspase-3-7 activation. And this indicates apoptosis via these pathways. But we did see that a minority were shown to act via caspase independent mechanisms, where they have this low percentage of caspase but are annexin-5 positive. And we've got some examples shown at the bottom here. For example, we can look at the different mechanisms between camptothecin and sertraline, with some example images from the AI cell health analysis shown for these compounds as well. The top 12 cytotoxic hits were identified, and these included inhibitors of topoisomerases, kinases, and 20S proteasome. A secondary assay was performed to calculate compound efficacy, in which the cells are treated with a 1 in 3 serodilution series of each compound in a 384-well plate. The percentage of dead cells over time was then quantified using the InKSight AI cell health analysis software module, and you can see the microplate view for this in A here. We also calculated the EC50 values using the percentage of dead cells at the 72-hour time point, as shown in the table on the right. So we also looked at the compound effect on cell cycle progression. In order to examine this, we used cells that are expressing the InKSight cell cycle reporter. The live cell population was classified according to orange and green fluorescence intensity, and this allows us to identify cells that are in the G1 phase of the cycle or in SG2M respectively. The kinetic analysis indicated there were different types of cell cycle disruption. So whilst mycophenolic acid caused cell cycle arrest in G1, as indicated by a consistent increase in the percentage of orange cells over time, flumazenil treatment caused the cell division to partially synchronize, with the time course displaying an increase in the amplitude of the percentage green cell peaks. And we've got some example images also shown of the effects here compared to vehicle. To examine effects on AK signaling, we quantified the inhibition of AKT in live cells by monitoring the translocation of a fluorescently tagged AKT substrate from the nucleus to the cytoplasm. During inhibition of AKT, this fluorescence accumulates in the nucleus. Using a correlation of nuclear fluorescence and cell morphology, this allows us to exclude compounds which induce cell rounding and are therefore given false positive results, and also enables us to easily identify true inhibitors of AKT. So we've got a scatter plot shown here showing the nuclear AKT substrate fluorescence and cell morphology for the cells treated with all compounds at 12 hours. And we've pulled out a couple of example compounds in the image grid shown in B as well. So we can look at the phase contrast and fluorescence images of vehicle, the AKT inhibitor AZD, as well as the auroronopin, where we can see this kind of false positive effect with the rounding up of cells. So to summarize, live cell imaging and analysis yields a multitude of valuable quantitative information, and this can be used to determine mechanisms of action and identify specific hits within phenotypic screening assays. Label-free data on cell growth and viability was extracted using the InKSight AI cell health and AI confluence analyses, and rapidly separated cytotoxic from cytostatic compounds. The inclusion of apoptotic dyes, including the annexin-5 and caspase 3,7 reagents, further determined the behavior of cytotoxic compounds by enabling the identification of caspase-dependent and independent apoptosis. Compounds affecting proliferation but not viability often act via targeting the cell cycle or signaling pathways such as AKT. The use of cells expressing fluorescent reporter lines were used to further examine these mechanisms. Kinetic analysis defined two groups of cell cycle disruption, arrest, and increased synchronization behavior, while the combination of morphology and fluorescence localization allowed us to identify true inhibitors of AKT signaling. Thank you for listening to this brief summary of the poster that we presented. Feel free to check out the poster itself, as well as other linked pieces of collateral, including webinars and application notes, which you'll find on our website.